Hello everyone, I hope you are enjoying this BES meeting online and I hope you are all doing fine wherever you are in the planet. My name is Frago and I work as researcher at CIRAT, which is a French institute for research in agriculture in tropical countries. And I will present you the results of an experiment that was performed in Reunion Island, which is a small island that belongs to France and which is located near Madagascar, even if currently I am based in Montpellier in the south of France. So the study I want to talk to you about deals on the effect of natural enemy diversity on herbivore suppression and community stability. This project was part of Karim Tihiward PhD, which was supervised by Samuel Nibush and myself, and which was in close collaboration with Dirk Sanders at the University of Exeter. So one of the main questions in this study is to explore the relationship between natural enemy diversity and herbivore suppression. This question has been studied for quite some time, and a recent meta-analysis has collected data from all around the planet, as you can see in the figure, and this meta-analysis has shown a very clear and strong positive correlation between natural enemy richness and pest and suppression on herbivores. This correlation is quite important because we know that natural enemies regulate herbivore populations, and these herbivore populations feed on primary producers, which are plants. So understanding this relationship is quite important to understand the regulation of terrestrial ecosystems. In terms of biocontrol and pest control, this relationship is also important because biocontrol practitioners always want to know if it's better to have two natural enemies or one or more in their agricultural ecosystems. So if this relationship is so positive, why we have been discussing about it for so long? Well, probably one of the main reasons is that when there's a lot of natural enemies in an ecosystem, these natural enemies can engage in antagonistic interactions among them, which I depicted some of them in this figure. For example, we have here an aphid, for example, that is feeding on a plant. This aphid is attacked by a parasitic wasp. Parasitic wasps are little wasps that lay the egg inside the insect, and then this egg develops into a larva that kills the insect. So this wasp is feeding on the aphid, but the ladybird is also feeding on the aphid, but at the same time, it can feed on the eggs that the parasitoid has laid inside the aphid. So this is an antagonistic interaction among natural enemies that is called intra-guild predation. Other antagonistic interactions can involve hyperparasitism, like a parasitoid that attacks another parasitoid, or top predators, which are predators that feed preferentially on other natural enemies. So the second question we were interested in this project was the role of natural enemy diversity on community stability. So community stability can be defined in many different ways, but probably the easiest one is to think about the persistence of the different species. If you have a community where all species persist through time, we can consider this community as quite stable. And of course, if all these species are maintained, this is good for maintaining diversity and to maintain the services that all this diversity can provide. We can also estimate stability by measuring the variations in density around the mean of the different species. And we can measure that, for example, with the coefficient of variation of these different densities of a given species. So there's been a lot of theoretical work on this relationship between stability and community and natural enemy diversity, and the results are not clear, and they are not giving us a clear answer to whether there's a positive or negative relationship. There's also quite some theoretical work on intra-guild predation, which suggests that intra-guild predation is only stable under certain conditions. So if all this theoretical work is telling us that the conditions for this maintenance of natural enemy diversity are quite rare, but in fact, we find quite a lot of diversity of natural enemies in natural ecosystems, and many of these natural enemies engage in intra-guild predation. Why is there this discrepancy between what the model says and what we find in nature? So this is why we thought about performing an experimental study where we had a community that we could manipulate to explore these questions. So the different questions we asked in our study is, does a larger number of predatory links increase herbivore suppression and community stability? And the second question was, does a larger number of intraguild predation links reduce herbivore suppression and stability? I will abbreviate intraguild predation as IGP, sorry, for at some points in this talk. And the third question was, what is the relative influence of predatory versus intraguild predation links on herbivore suppression and community stability? So to explore these questions, we used quite a complex community composed of 8 species for herbivores and for predators. I depicted with arrows the the predatory links, and in red I depicted the intraguild predation links. So you can see that we have 4 herbivores at the bottom of this slide, 2 thrips, 1 spider mite, and 1 aphid. Then we have 1 predatory mite that feeds on the thrips. Then we have 1 parasitoid and 1 ladybird that feed on aphids. The ladybird can also act as an intraguild predator of the aphid. And then we have a quite generalistic predatory bug that feeds on almost everyone. In total, this community has 11 predatory links and 3 of them are intraguild. So using this community, we designed 8 different communities by removing 1 or 2 of these natural enemies. We maintained always the 4 herbivores, and we created a kind of a gradient of the number of predatory links and also gradient on the number of intraguild predation links. Of course, these 2 variables are quite correlated, but when we design these communities, we try to reduce this correlation as much as we could. Just a small example of how we develop these communities. We have here the full community. If we remove 1 species, for example, the ladybird or the predatory bug, we end up with communities with a variable number of predatory links and intraguild predation links. We set up the experiment in microcosm cages that you can see in the picture. We replicated these 8 community types 10 times, so we ended up with a total of 80 microcosm cages. These insects, the herbivores fed on beans and tobacco, and we selected the species, this community, because all the species have short generation times. So we followed the dynamics of these communities for over 20 weeks. And since these species reproduce quite quickly, we managed to follow quite some generations of these different species, so we can talk about long-term dynamics, stability of the system, persistence, extinctions, and stability in general. So this is the type of results that we obtained. You can see here the number of aphids through time, and the different community, the different colors are the different community types that we deployed. And for each cage and each species, for this talk that I will present today, which are quite preliminary results, we just obtained the cumulative number of insects for each species and for each cage. With this temporal dynamics, we also estimated the coefficient of variation as a measure of stability. And then we also estimated persistence, species persistence, and the time to extinction. So for example, in terms of the time to extinction, you can see here the dynamics of this aphid. So the different points, of course, are different time points, and the variation around the mean is the variation associated to these 10 replicates. You can see that most of the communities, they have a peak and then they go down, then they have a second peak. But for example, there's few communities here that from the week 15 on, they lay flat on the 0 because the aphid became extinct. So for each case, we calculated at which time these extinctions occurred, and we obtained the extinction time. So based on this data, we modeled a few mixed effects models where we included as explanatory variables the number of predatory links and the number of intraguild predation links. And we included as a random factor community type to correct for any variation associated to the community type, of the uniqueness of this community type. For example, we didn't include in our analysis the nestedness of the different community types. And by including this random factor, we corrected for this unique variation associated to the type of community among these 10 different replicates. So if we take a look at the results on the effects of these different variables on herbivore density, the first result is that integral predation links had never an effect on herbivore numbers. However, predatory links related significantly with the numbers and survival of 3 out of the 4 species, but on different directions. For Echinothrips americanus, for example, one of the 2 thrips, we found that the cumulative number of thrips on these cages related negatively with the number of predatory links. So more predators, less thrips. And we found the same for the aphid Myzus persicii. And for Echinothrips americanus, we found also a negative effect on survival time, which is quite associated to the density. If you have a community of natural enemies that suppresses a lot of density, it's quite likely that this will also trigger extinctions. But for the spider mite, we found a positive effect on the number of predatory links on cumulative numbers of these species. So this suggests that more species of natural enemies may release the populations of these species. In terms of the effect of these different variables on the coefficient of variation, again, we found that integral predation links had no effect on the coefficient of variation, but they affected, the coefficient of variation was affected by the number of predatory links, but again, on different directions. For the spider mite, we found that a larger number of predatory links reduce the coefficient of variation. So more diverse community of natural enemies made the populations of this mite more stable through time with less variation. And we found the opposite for the aphid Myzus persicii. So to wrap up a little bit based on the 3 questions that we proposed, we found that clearly the number of predatory links had an effect on herbivore suppression and community stability, but this was variable depending on the species, and not all species were equally affected. The number of integral predation links were not affecting at all the dynamics of our system, and clearly predatory links are stronger predictors of the dynamics in our system than integral predation links. So the analyses I just showed you are quite preliminary, and I would like to finish by showing you a bit of a more complex model, a path analysis where we included all the data from all the different herbivores and the different natural enemies. Here you can see again the full community with the different trophic links, and what this path analysis has shown us is that, for example, it had an impact on the 2 species of thrips, but the impact was negative on the black thrips, but positive on the other. So probably what's happening here is that this predatory bug has a preference for the black aphid, and this is relaxing intraspecific competition between these 2 thrips. We also see a negative impact of the predatory bug on the ladybird, probably because they compete for the aphid resource. If we take a look at the time series data, for example, for the parasitoid Aphidius ervi, we can see here I just selected a couple of communities which are exactly the same and that only differ by the presence or absence of the ladybird. And we can see that in the red community, in these parasitoid numbers, where the ladybird is absent, we have a big peak of parasitoids and then the parasitoid becomes extinct. Whereas the blue community, where the ladybird is present, the density is a bit, the peak is less dramatic, and then it seems that the parasitoid persists for a bit longer. If we take a look at the other graph, which is a graph of the survival of this parasitoid, the number 1 means that at the beginning of the experiment, all cages contain this parasitoid, and through time, the species becomes extinct in some of these cages. So we can see here clearly that the presence of the ladybird is allowing this parasitoid to persist for longer. So maybe intravenous predation ultimately can be beneficial for the persistence of some species. And that's all. I just wanted to thank again all the people involved in this study. I would like to thank especially La Coccinelle, which is a company that provides natural enemies in Reunion and who was the company that was providing us with all these nice natural enemies. And thank you everyone for listening and enjoy the rest of the meeting. Bye.